Hello everyone. Today we will discuss about hedgehog pathway, right? It is mainly responsible for cell proliferation. Now the important thing here to understand is that in inactive state, basically when there is no signal, the target genes are silent. But once you get your hedgehog, the target genes turn to be active. The main signal is CI protein, right? The cleaved CI act as a co-repressor which silences your gene. This hedgehog helps in, intact to not be cleaved, and therefore the target genes remain active. Now the components are ligand, that is hedgehog, the receptor is patched and ihog. Now there is a transducer also, which is smoothened. Very important in the signaling, okay? So this is very similar to GPCR. And there are scaffold proteins. One is causal 2, the other is fused. And the effector is cubitus interruptus. Now, the thing is that this complex is degraded with the help of, so you can see that this is the scaffold protein. So the CI protein is basically bound in it. And this is basically phosphorylated, right? The cleaved one with the help of these three kinases, which is also similar in your Wnt-catenin pathway, right? So this is later on proteolytically cleaved and that act as a transcriptional repressor in the nucleus. Now let us see the diagram first. Okay. So what happens without the hedgehog signal? So there is patched, there is ihog. Okay, so in absence of signal, they both are separated. But this patch, we don't know the exact mechanism, but it represses the smoothened so that it remains inactive. So it is inside the vesicle in an inactive form. Now, this large CI protein is basically in general without the signal is in bound form with the scaffold protein that is costal 2, which is somehow connected with the microtubule and the other protein which is fused. So what happens is later on there is phosphorylation of CI1, CI by these pKa, GSK3 and CK1. And later on this causes ubiquitinylation and proteolytic processing, and therefore this cleaved CI protein gets into the nucleus and recruits co-repressor. Therefore their hedgehog target gene is off. Now, with hedgehog, it's very important to understand. So this is your hedgehog. This comes in between the ihog and patch. This causes this patch to be degraded. I am degrading this patch in lysosome. First you internalize this patch, then you degrade in lysosome. So you don't have patch. So this smoothened, which is inactive, is now active. Comes out of the knee, comes out of the vesicle and sits here. Now, this phosphorylated smoothened will destroy this complex and will free this CI. So basically, this smoothened helped in inhibiting CI proteolysis, right? So therefore, the CI remains active, goes in, recruits a co-activator, and therefore your hedgehog signal remains active. Now, this is your negative feedback and auto-regulation, how it's basically done. So, what is the negative feedback that you can imagine, right? That is, if I don't want this feedback, then I will just say that there should not be any signal or there should be patch receptor so that it can keep the smoothened in inactive state. So this is your negative pathway. So what happens if you have CI, intact CI a lot, right? It will go and help in patch gene activation, and therefore newly synthesized patch receptors would be there, which will therefore inhibit the pathway, right? It's very important, right? Because if there is a lot of cell proliferation, or if there is no proper signaling, then it can be cancer causing, right? We will discuss it. Now, the important thing is that there is an evolutionary divergence as well. In Drosophila, you have hedgehog. Here, the ligands are sonic desert and Indian hedgehog. Here, the CI protein is there. Here it is GLY, GL2, GLI3. So, similar mechanism, right? It's in microtubules, it is in primary cilia. Okay. Now, this is very important to understand. Tipping the scale, right? Now, what if this small molecule is being this hedgehog signal is in a turned off state, right? What will happen? There would be severe developmental defects. There would be severe developmental defect. So how? We had this is called a very potent compound, which is cyclopamine. It is a very small molecule found in plant meadow lily. So what happens is that the sheep which was gazing on the meadow lily produced progenies with cyclopia, means a single eye, due to missing morphogen signals. So can it be useful to reduce hyperactive signals? Definitely, this compound can be useful. Now, the next thing, what if there is hyperactivation? Right? Suppose if I inactivate patch the gene, right? So smoothen will be free. It won't be in inactive state. It won't be in the vesicle. Therefore, resulting in excess, there would be too much of signaling. So there would be basal cell carcinoma, one of the most common form of cancer in Caucasians. So obviously it must be right, perfectly balanced, because if there is very little, then the embryonic development will be derailed, right? If there is too much, then you can have uncontrolled oncogenesis. Now, here is the just I have written down like few important similarities, which is Wnt and hedgehog. They both act as local mediators. They have shared protein kinases. These signal proteins are modified by covalently attached lipids. Now, both pathway trigger a switch from transcriptional, repression to transcriptional activation, right? And this gene was discovered in Drosophila when the mutation in the hedgehog protein produced spiky projections called denticles, like this in the world. Okay, thank you.